This is a production of Cornell University.
Okay. So the title of my work is Evolution and Development of Ecosystem Models to Understand Carbon and Hydrological Processes. So as we all know, soil, it is the largest reservoir in the terrestrial ecosystem, which is more than the vegetation and atmosphere carbon dioxide combined. And even a small change in soil carbon stock can influence a big impact on climate or ecosystem. So what is really happening in soil? We want to understand. To understand it, we use process-based models, and there are several process-based models being developed to explain what is really happening in the soil, for like decomposition, soil organic carbon decomposition. But still, lots of uncertainties still exist. As we can see here in the graph, no Earth system models are able to accurately estimate or predict soil organic carbon in the terrestrial ecosystem. So, to explain, to address these uncertainty, I studied couple of processes in the terrestrial ecosystem, like microbial models and hydraulic redistribution. My first chapter was a review work, which I reviewed more than 70 microbial models. And the next two chapters, I studied hydraulic redistribution, which I'm very excited to present today. So first, microbial models. So what is a microbial model? When we compare it with conventional model, they don't have a microbial biomass pool here, as we can see. But in microbial models, they are explicit representation of microbial models, microbial biomass. And in the recent decades, because of the advancement of microbiology, we can see there is a recent very high number of development of microbial models. Yeah, in latest 2000th century, we have more than 60 microbial models developed. So it was very important to review what is happening in the microbial models scenario. So to put forward, I developed these objectives, that is to identify and characterize the major microbial processes incorporated in microbial models, to elucidate the environmental factors influencing microbial processes, and to identify the challenges associated with microbial models. So after reviewing more than 70 models, I found that there are four major microbial processes that has been incorporated in microbial models. That is microbial mediated decomposition, mineral interaction, microbial necromass recycling, and active and dormant microbial dynamics. One by one, I would like to explain all these microbial processes. Uh, the first major process is microbial mediated decomposition of soil organic carbon. There are several mathematical equations how these microbial mediated decompositions have been represented in microbial models. I'm trying to explain here. Uh, like zero order, the first one is zero order microbial model. As the name suggests, even there is a representation of microbial biomass in the model. This biomass does not actively participate in the decomposition equation. So the decomposition rate does not depend on microbial biomass. So I named it zero order microbial model. Then, then the next one, first order microbial model. Here, the microbial biomass was directly related with the decompositions as a linear fashion. So the decomposition was linearly related. So I named it as first order microbial model. Then there are several non-linear microbial model that are represented by forward Michaelis-Menten and reverse Michaelis-Menten. In the forward Michaelis-Menten, uh, the decomposition is limited by substrate, as here represents the amount of soil organic carbon present in the soil. So the decomposition rate, it is limited, rate limited by substrate, whereas in reverse Michaelis-Menten, the decomposition is rate limited by enzyme, microbial enzyme. Uh, then combining all together, most of the microbial models, they are represented by non-linear uh, equation. So like here we can see most of them are represented by non-linear, and only few models were represented by linear models, either first order or zero order model. So what in the conventional model again, um, conventional model assumes that the the decomposition of any soil organic carbon, it is not limited by microbial biomass, and they implicitly assume that the decomposition of carbon substrate um, is different than like active soil carbon pool, slow carbon soil carbon pool, and passive soil carbon pool. So these are conceptual biomass, soil biomass, yeah, soil carbon pool in the soil. Uh, those have different decomposition rate, but in microbial biomass, the soil organic carbon, they have different decomposition rate because some soil organic carbon are attached to microbial, attached to mineral component, and they are not actively available for decomposition. That's why they are not taking part in the decomposition. Then if there is a microbial biomass in the model, what is happening with the microbial biomass? In some microbial model, they pushed forward. They added, sorry, uh, they added one more carbon pool, that is microbial necromass, that contains dead microbial biomass and also enzymes secreted by microbial biomass. Because these microbial necromass, they have different decomposition rate than the plant residue or other soil organic carbon pool because of different carbon nitrogen ratio. Then some models also assume that some microbes, they are smart. When the environment is not favorable, they can go into dormant phase. And these dormant microbial biomass, they will not actively participate in decomposition. And when we have favorable environmental condition, they again switch to active microbial biomass and start to decompose soil organic carbon. Uh, in microbial model, these processes are also influenced and affected by environmental controls. Most of the models, they use three main environmental controls: soil temperature, moisture, and pH, with varying uh, functions to represent in the model. But even these are very like uh, microbiology based microbial model development. Still there are several challenges associated with microbial models. Like first one, microbial model core biggest assumption is that the decomposition of soil organic carbon, it is limited by microbial biomass. But some studies, they suggest that that some organic carbon, they are rate limited by abiotic factors like mineral in the soil. So we need some experiment to prove that the decomposition of soil organic carbon is limited by microbial biomass, and more observation data is needed to constrain more microbial models parameter. And the third, incorporation of more processes makes the model too complex to constrain parameters. So in conclusion, I found these four processes: microbial mediated decomposition, mineral interaction, microbial necromass, active and dormant physiological states. And um, like even these four processes, they are represented by several mathematical equations. It means same processes, we have very difficulty, and there are uncertainty in translating these knowledge into model. And the biggest recommendation is we need data-driven model development and parameterization for highly recommended to improve prediction of microbial models. So now next two chapters, I'm very excited to present my modeling work. That is hydraulic redistribution, chapter 2 and chapter 3. In the chapter 2, I will discuss about hydraulic redistribution, decreases with precipitation, magnitude and frequency in a dry land ecosystem. It is a data model fusion approach. So first, what is hydraulic redistribution? Um, it is a vertical movement of water among soil layers by root from wet to dry soil layers at night time mostly. driven by water potential gradient. Water can move from deep soil layer where we have more water to shallow soil layers at night time, preferably, mostly at night time when we don't have transpiration. And downward hydraulic redistribution can happen when there is a rainfall and top soil layer have more water than the deep soil layers. So downward hydraulic redistribution can happen. So it is not a conceptual based process. It is really identified in the field. It can be seen by using root sap flux measurement, where we install some sensors in the lateral roots, and we detect water is moving from away from root to the soil. So these negative lines below zero, they represent that water is moving away from the root. That we found the presence of hydraulic redistribution in our study site. But this is one tree location. The magnitude of hydraulic redistribution is still uncertain, and there has been several studies conducted, and still magnitude is a debatable topic. So to improve hydraulic redistribution representation in model, uh, I used one a classic hydraulic redistribution model to my process based ecosystem model, that is TICO, which I will explain. The main purpose of this study was to realistic implementation of hydraulic redistribution in a model. So for the objectives of this study was to incorporate hydraulic redistribution process into an ecosystem model constrained by observational data through data assimilation technique, and then to identify the key mechanisms determining the presence and magnitude of hydraulic redistribution, then to quantify the dynamics and of hydraulic redistribution across multiple soil depths. So this is some information about the study site. The mean annual temperature was 10.5 degrees Celsius, annual precipitation 170 to 400 millimeter. The soil type is sandy loam, and the vegetation mostly 60 to 80% is juniper pinyon in our study site. So to implement a realistic process in a model, we need these three steps to fulfill. That is, first, structure. We should have a correct representation of equation in our model. That I added hydraulic redistribution, a process in our ecosystem model. Then parameterization, I use data assimilation technique to optimize parameters to represent our study site, and external forcing like precipitation, temperature for the realistic projection. So this is the TICO model, that is terrestrial ecosystem model. It represents an ecosystem for a field study site. It has all the components like canopy, model, photosynthesis where it's happening, uh, photosynthesis transferring to leaf, stem, and roots, then these biomass going into the soil, uh, litter formation, and then we also have soil water dynamics. So I added hydraulic redistribution in our soil water dynamics module. The soil water module was balanced by unsaturated water flow, evaporation, transpiration, and hydraulic redistribution. This is very important. Was unsaturated water flow, how water usually flow in the soil. This is represented by Darcy equation, or also known as Richards equation, where water moves because of this water potential gradient, and plus one here represents the gravity. So water is moving among the soil layers, like deep in the soil layer, through two processes. Based on water potential difference, it can move upward and downward, and plus one, it will always go downward through gravity. So, and this K represents hydraulic conductivity, which was represented again by Brooks and Corey model. Um, the red color coded parameters, I used data assimilation to optimize these parameters using soil water content data I had from the field. Then this process, all about this process, hydraulic redistribution model. It is represented by CRT, that is maximum radial soil, how much plant root is allocated in the soil layers. Then it is difference between water potential gradient between the soil layers. Maximum, the CI represents relative soil root conductance, how much soil and root they are connected with each other. And RI, RJ, they are the root fractions available in each soil layers. DTran, it is a switch, because hydraulic redistribution, it happens only night time. So this DTran has two values, 0 or 1. At night time, it will become 1. At daytime, it will be 0. And this is controlled by solar radiation. So if there is a solar radiation, it will the model will assume it is a daytime. If not, then nighttime. So this is the model result. The black line, it represents observation. Blue, it is TICO plus hydraulic redistribution, that is modified model. And default TICO means without adding hydraulic redistribution. Here we don't see much improvement, but if we see more closely, we can see the blue line, that is a modified model. It is aligning well with the observation. Um, and also hydraulic redistribution model is not active all the time. We need to find out what time period, at what environmental condition it is active. So if we see RMSE error here, the first column it represents the RMSE for whole time period. Not much improvement, little bit improvement, but not much. Mostly at dry period, we see a major improvement in model performance for for volumetric water content. And again, wet period, uh, not much improvement, because this dry land does not have very high volume of rainfall. So water is not moving, like hydraulic redistribution is not contributing much, moving water from topsoil to deep soil layer. But because of dry land ecosystem, water is moving from deep soil layer to topsoil layers. That what we expected here. We can see here in dry period, we have improved model prediction. This is a total model simulation. Here we can see diurnal HR simulation, hydraulic redistribution simulation. One note I would like to say, TICO model, it has 10 soil layers. So here it represents 10 soil layers from 5 centimeter to 180 centimeter. And this is the representation how water is moving, this is how hydraulic redistribution is moving water from deep soil layer to top soil layer. So when we don't have precipitation, water is moving from deep soil layer to top soil layer. These lines, they are the top soil layers of 5, 15, and 30 centimeter. And at the same time, we are seeing the deeper soil layers, they are losing water. So it also represents the water mass balance. Similarly here, when we have precipitation, uh, very little amount of precipitation. Still, hydraulic redistribution is moving water from top soil layer to deeper soil layer. That is shown in negative direction. And the like 30 centimeter soil layer, it shows some increase in water content in hydraulic redistribution, which represents deep soil layer, means top soil layer is moving to a deeper soil layer because of precipitation. Here we see daily hydraulic redistribution simulation at different soil depth, mostly at 5, 15, 30, 60 centimeter, which I had data to compare with. So here we can see when we have precipitation, hydraulic redistribution is coming close to 0. And when we have dry, water is like hydraulic redistribution getting in the very positive side. Water is being moved from deep to top soil layers. So again, we wanted to know how this, how hydraulic redistribution is working in the ecosystem. So we found a significant correlation between precipitation and hydraulic redistribution magnitude. In the topsoil layers, we see there is a decrease. There is a negative relationship between hydraulic redistribution and precipitation. But in the deep soil layer, when we have much precipitation, high volume of precipitation, deep soil layer hydraulic redistribution is getting positive, which means uh, at high precipitation it is gaining water from the top soil layers. And not only precipitation, the drought period at the in the environment also controls how controls the magnitude of hydraulic redistribution. So the x-axis it represents dry spell length, how long the dry period was in that between two precipitation events. So if we have longer dry spell between two precipitation events, we have high hydraulic redistribution. What means more water is moving from deep soil layer to top soil layers. Then because this water is moving, so we wanted to see how it is influencing overall soil moisture. So when we have dry condition, persistent dry condition, water is getting, so a topsoil layer is gaining more water. This is a heat map and, graph. So at persistent dry condition, we see increase in soil moisture up to 30 centimeter in topsoil layers. But if we go deeper in the soil, this volume is decreasing. So the conclusion of this chapter was hydraulic redistribution improved soil moisture prediction, especially in the top 30 centimeter with greatest impact during dry periods. Surface soil moisture increased by up to 30% during dry periods. Hydraulic redistribution decreased with increasing precipitation magnitude and frequency, but was also influenced by length of dry spells between rainfall events. In this ecosystem, because of dry land ecosystem, we have upward hydraulic redistribution in top 60 centimeter. Soil profile became more pronounced as dry condition progressed, with rates ranging from 0.03 to zero point 0.2 millimeter per day. Then the third and final chapter, that is investigating the influence of hydraulic redistribution on carbon and water dynamics. The overall objective of this project was to quantify how much hydraulic redistribution is influencing on carbon and water dynamics of ecosystem. So to conduct this study, I used data from two study sites. One is from US MPJ New Mexico site. It is a Ameriflux site where we had data for soil water content at 5, 10, 30 centimeter, and NEE, net ecosystem exchange data, to compare and validate our model. Again, this ecosystem also similar, pinyon and juniper, sandy soil, 385 millimeter average precipitation and 10.5 average temperature. Whereas the second site, alpine meadow, it is a grassland ecosystem having mean annual temperature 1.5, annual precipitation 760, different type of soil, loam soil, vegetation, yeah, again grassland. We have soil water content at 5, 10, 20, and 40 centimeter. So the model was calibrated using soil water content and net ecosystem using data assimilation. So this is very interesting finding from the both. If we compare both sides, the first one is still dryland ecosystem, but we have very frequent precipitation, not very seasonal. Throughout the year, we have a small amount volume of precipitation throughout the year. So we did not see much hydraulic redistribution in this area. Uh, So hydraulic, mostly it was still upward hydraulic redistribution because it is positive, but very minimal, 0 to 0.5 millimeter water per day moving from deep to top soil layer. Whereas this alpine meadow, here we can see we have seasonal precipitation only mostly at the growing period. So we see a significant negative hydraulic redistribution, means water is moving from top soil layers to deep soil layers. That is downward hydraulic redistribution. So again, if we compare model result with observation at US MPJ site, we don't see much effect in NEE data, NEE simulation, model simulation with and without hydraulic redistribution. Red color line is modified model with hydraulic redistribution, blue is without. We see little bit change, but not much substantial. So We also see some improvement in model simulation when we added hydraulic redistribution, but again, not much. Hydraulic redistribution decreased carbon dioxide uptake from atmosphere during April to June by 0.03 to 0.2 gram carbon per meter square per day, in average zero point 0.1 gram. Uh, yeah, so at this site, because of very frequent precipitation, we did not see much effect of hydraulic redistribution happening in this site. But if we see second site, Alpine Meadow, we see some difference in with and without hydraulic redistribution. Um, After adding hydraulic redistribution in the model, we saw a little bit improvement, overall improvement in model simulation R square value, 0.44 to 0.48. But if we see at particular time period, because hydraulic redistribution does not work all the time throughout the year, we need some particular time when it is most prominent in the ecosystem. So I found these two time periods. So this here we see hydraulic redistribution decreased carbon dioxide uptake from atmosphere during April to June, that is early growing season, by 0.07 to 0.53 gram carbon per meter square per day. If we compare with the hydraulic redistribution, because water is moving from topsoil to deep soil layers, so hydraulic redistribution is reducing water content in the topsoil layers. That is the reason we see reduced carbon dioxide uptake. But if we see here, there was a gap. Even in this growing period, if we have a gap of precipitation, we see model uh, during late growing season, hydraulic redistribution increased carbon dioxide uptake by 0.4 to 0.31 gram per meter square um, per day, because we can see there is reduced in downward hydraulic redistribution. Water, there is not much change in water. Yeah. So again, we wanted to see how hydraulic redistribution is affecting water dynamics of the ecosystem. At US MPJ site, we did not see much influence of hydraulic redistribution, but at Alpine Meadow, we see at early growing season, there is a reduce in available soil moisture because of downward hydraulic redistribution, and then hydraulic redistribution reduced ET, that is evapotranspiration. Here red line represents modified model, that is TCO plus HR. Um, yeah, it is matching with the data, and hydraulic redistribution reduced ET by 0.7 millimeter water per day. So the conclusion of this chapter was to no significant hydraulic redistribution and its influence on ecosystem functions was detected at US MPJ site, likely due to two main reasons. Because of frequent precipitation, there is no prolonged dry period, so hydraulic redistribution did not get activated much. And also because of sandy soil. Sandy soils, the water moves very fast through the soil profile. So there was not much retention of water in the sandy soil. But at the alpine meadow, due to seasonal precipitation, model predictions suggest uh, downward hydraulic redistribution of magnitude of up to 0.22 millimeter water per day, and decrease in carbon dioxide uptake ranging from 0.03 to 0.32 gram carbon, and it also reduced at some time period ET up to zero point 0.7 millimeter water per day. Uh, and the take home message from all of my three chapters, like first chapter, it is microbial models. The biggest recommendation or suggestion was to data driven model development and parameterization are highly recommended to improve the prediction of microbial models. From second and third chapters, uh, precipitation pattern and soil texture are the stronger drivers of hydraulic redistribution than biome itself, because we saw a same kind of biome, precipitation is influencing hydraulic redistribution, and then the incorporation of hydraulic redistribution could improve somewhat little prediction of ecosystem functions at extreme environmental conditions. Yeah, I would like to thank my advisor and committee, Dr. Lu, Dr. Sangtao Su, and Dr. Scott Steinsnyder, Dr. Lifen Jiang, and all my collaborators and friends. Yeah. If we have time, yeah, I'm happy to take questions. Thank you. Yeah.
Thank you, Aneesh. I was wondering mostly regarding your second chapter. So how would you say, if we think about the potential for climate change to have longer dry periods and potentially having those, droughts that are for longer. So how would you say that I mean, I guess it would increase the importance of including the hydraulic redistribution in models, but how do you think that would be likely to affect anything?
So your question is how it will affect when we have climate change, a longer drought period.
Yeah, besides that, we should maybe
Yeah.
include it more in models. Like, it seems like more important than to include.
Means, yeah, if we have more drought period, I think that is beneficial to add hydraulic redistribution, because as we have more drought period, as we go further in the dry condition, the hydraulic redistribution is more activated. So it is adding more water from deep soil layer to top soil layers.
Okay.
So it is only beneficial we have really longer dry period. And also if we have short dry period, adding it, is it not also harmful? It is also matching well without adding hydraulic redistribution. So adding is not harmful,
It's
and
not bad,
yeah,
but it could
yeah,
help when it's drier.
yes,
Okay, yeah, thank
yeah,
you. Yeah.
yeah.
So, Anisha, I probably asked you before, do you have soil moisture data? Does top layer soil moisture increase at nighttime in the observational data? If hydraulic distribution has a big impact, we should see that signal.
So in the soil moisture, as other literature says, there should be increase in soil moisture exactly at night time. We do see amplitude like increase in the like 24 time period. We see increase and then decrease of soil moisture, but not exactly at night time. In our data, there is some lag, time lag, which shows, um, yeah. But in the hydraulic redistribution, we assume it happens mostly at night time, but there is some lag. Soil moisture is getting highest at 11 a.m., 12, like that. But we do see there is a amplitude fluctuation of soil moisture. Yeah, without rainfall in dry condition. Yeah, yeah.
I just had a question about your perspective on chapter 1. So the Michaelis-Menten style models, when they're looking at microbial biomass, they're usually substrate driven. So the activity is sort of dependent on the substrate concentration. From an ecological point of view, we describe that as bottom up. So you're limited by the substrate. But there's another model where you could be top down, where the growth of the activity is driven by consumption by like a predator or a virus, ciliates, nematodes. So in a top down system, you are sort of uncoupled from Michaelis-Menten kinetics, because now it's the rate of consumption and the microbes grow to keep up. So it could change the efficiency and then change basically the amount of necromass that you get. Did you encounter any models, or were there any sort of recognition of this top-down versus bottom-up issue in the models that you explored?
So yeah, unfortunately, no, I did not see uh, this. Maybe it is more microbiology side paper I have to review. This was more ecological side, pure modeling works. So I think more time we will need. Modeling side, they need more time to incorporate those understanding.
My sense is that they're mostly based on Michaelis-Menten, but
Yeah.
you've read many more papers on this topic than I have,
Yeah.
so I value your opinion.
Yeah.
Thank you.
Yeah. Thank you.
Great. Thank you so much.
Thank you.
And thank you all.
This has been a production of Cornell University, on the web at cornell.edu.